Hi, this is Allie Denton and you're watching my exam mini video 3 for Genetics 311. Today we're going to be talking about the synapsis of chromosomes. The synapsis of chromosomes is when two homologous chromosomes pair up in meiosis prophase 1 and cross over genetic information which results in genetic variation. We can take a closer look at the synapsis of chromosomes in individuals. We have two chromosomes from two individuals. The first individual has MLI centromere JKHG and GHI centromere JKLM. The second has MLI centromere JKHG and GHKJ centromere ILM. While looking at individual one's chromosomes, we want the chromosomes to match up as best we can, so we can flip the second or the first one around so it lines up just a little bit better. In order for these to match up, we would need to create an inversion loop. There are two types of inversion, paracentric and pericentric. We can tell that this is a pericentric inversion because the flip contains the centromere. Inversions without the centromere are called paracentric inversions. Now moving on, we can draw out a picture of our inversion loop. We want all the letters to match up as best we could, so with the bottom two lines we created a loop. Let's take a look and see what happens with the chromosome if there was a crossover between the J and K regions. First, we trace along each line along the lines drawn, including the switch. The yellow line looks pretty normal because it lines up exactly with one of the original chromosomes. Next, we trace the second line in pink. This one looks slightly different because there is a crossover at J and K, causing some of the letters to repeat, giving us GHI centromere JKHG. The third line we trace in blue. This one also looks identical to one of the original chromosomes. Lastly, we trace the last chromosome in orange. It starts on the right side and ends on the right side as well, after crossing over in the JK region. This gives us MLKJ centromere ILM. How do we know which would produce viable gametes or not? So to produce viable gametes, the chromatids must not repeat or skip any letters. We can tell from the letters in the yellow that it never repeated and all the letters were used, so this one is viable. Pink repeats, so it is not. Blue is viable because it contains all the letters and does not repeat, and orange is not viable since it repeats and doesn't include some letters. Individual 2 has these chromosomes. If you swap them around, you can see clearer which lines up with what. After swapping the chromosomes around, we can tell that these are actually identical to each other. Looking at the two chromosomes from individual 2, we can see that both of their lines are parallel to each other. And we're going to take a look and to see what happens if we do a crossover at the J and K regions. While doing the crossover at the J and K region, we find that we are getting the same thing each time. Viable gametes are chromosomes that do not skip or repeat any letters. Since all of these are the same, we only need to look at one and we can tell that none of them skip or repeat letters, so they are all viable.